Okay. All right. You recap questions? I forgot my field. I was coming from another seminar. Okay, so why is it important to analyze protein content? Remember? For nutrition and nutrition labeling. 3. Functional properties of the protein are always estimated by gram protein. Sally, quality control. For quality control and standard of identity. Okay. No, Eudora. Yeah, biological activity. So if it's an enzyme or if it's a bioactive peptide, we need to determine the amount of protein so we can determine activity per amount of protein. One last thing, page.
Protein fractionation.
Protein fractionation and production of protein ingredients to determine the purity of the protein. Great. Why might the nitrogen conversion factor be different for different sources of proteins? Who remembers? Go ahead, Tyler.
Proteins are made up of different amino acids, and each amino acid has
Different amino acids have different nitrogen composition, especially those basic amino acids that have extra amino groups. They have different composition. So if you have a protein that is high in basic amino acids, your conversion factor will be lower than a protein that has low amounts of basic amino acids. Okay. What are the 3 main reactions, chemical reactions in Kjeldahl? Let's see I'm going to go with Maya. Digestion, distillation, and titration. That is correct. We're going to review all of them again, just to refresh your memory what happens during digestion, what happens during neutralization / distillation, and then what happens during titration. Okay. So during digestion, with a strong H2SO4, so concentrated sulfuric acid, what happens? Do you remember? When you digest with a strong acid, what happens to your organic matter? Sally? Combustion. So we have combustion, oxidation, that is, of carbon matter into CO2, into water vapor, into sulfur dioxide. But what happens to the amine groups, to the nitrogen? It gets fixed into ammonium sulfate. That's an important term. When I ask you in an exam, state the principle of Kjeldahl, you want to tell me what happens to the nitrogen at each step. The first step, the nitrogen is fixed as ammonium sulfate. It doesn't get evaporated. Gets fixed as ammonium sulfate. The next step, which is neutralization and distillation. In this step, you have this digested sample. Everything organic has combusted except the amine group was fixed, and this blue color because of the catalyst. We put in here sodium hydroxide, an amount that would neutralize the acid, but at the same time, ammonium sulfate is going to interact with sodium hydroxide to give you ammonia. Now your nitrogen is in the form of ammonia, which is a vapor, a gas. So what we do in this distillation unit is capture the ammonia. So your ammonia and water, because when you have ammonia, when you have sodium hydroxide reacting with sulfuric acid, and what happens is you have an exothermic reaction. So even water will evaporate, ammonia will evaporate, and then together will be condensed into a receiving flask that has boric acid. So your ammonia is going to react with boric acid, giving you ammonium borate. So now your nitrogen is in the form of ammonium borate, And in the presence of an indicator, which we will put the red indicator, like the red is yellow in the presence of a base, and it turns pink when it gets acidified. So what you do is you have your ammonium borate, you titrate with HCl, and then the last drop is going to turn your indicator red, which means this is neutralization. The next drop is going to acidify your solution, and it's going to turn the methyl red into pink color. And that's where your titration ends. This is the unit I have used when I was a master's student. It's a very old but functional unit. So here, the blocks is where you put your digestion flask. And up here is your distillation unit. But you have an exhaust that will take care of all the fumes, because you don't want to inhale the fumes. You want everything to be captured by the exhaust system. And we'll see in the lab, we have a different exhaust system that we use for the digestion. But this is the old style. And okay, so there is a story behind this. So when I was doing a lot of Kjeldahl during my master's, so this was a lab in the hallway where across from the labs are all of the offices of the professors. So one time, for some reason, I was doing the digestion and the exhaust system broke. So there was fume coming out of this and into the hallway. So somebody called me and said, there's something wrong. So I was running up and I got so scared because there was this cloud and there was this one professor that was very tall. So all I can see is the head over the cloud running at me and I said, what have you done? I'm like, I don't know. I didn't mean to. And it's like, this is a story I will not forget because I was really scared by just seeing a head over the cloud running at me. But fun times doing Kjeldahl. Anyway, so calculation. So once we titrate, what we care about is the volume of NaOH taken to reach neutralization. So I'm going to work with you some calculation to derive this equation. By understanding the derivation of this equation, you can probably hopefully remember it. So what happens at equilibrium? So at equilibrium, you have equivalent of one reactant equal the equivalent of another. So at equilibrium, basically, number of moles of HCl equal number of moles of nitrogen. I'm going to say nitrogen. Why? Because you have your HCl with your hydrogen reacting with the borate. And you have 1 mole of ammonia and 1 mole of borate. So in ammonium borate, you have 1 mole of ammonia. And in 1 mole of ammonia, you have 1 mole of nitrogen. So at equilibrium, number of moles used of the acid is equal number of moles of nitrogen. And what am I after is I'm after the mass of nitrogen so that I can get to the percent nitrogen, which I can use conversion factor to convert to protein. So I really need the mass of nitrogen. All right, so number of moles equals normality times volume times equivalent. Yeah.
Quick question. What is that equal sign with the little circle over it? Where it's N something?
Number of moles. Okay. This is how I do it. Usually I call number like this, or you can call it like this.
Oh, okay.
just number of moles, forget the way I write it. All right, so number of moles of HCl equal number of moles of nitrogen. So number of moles of HCl is the concentration times the volume. Okay, so basically N by V of HCl. Okay, so normality times volume. Since molarity equals normality because you have the one hydrogen. So number of equivalents is 1. So you can say N by V. All right. I'm going to make a change here I'm going to put N here, HCl here. Okay. And number of moles of nitrogen is going to be equal mass over molecular weight of nitrogen. Are we following? If there's anything I need to repeat, it's now that you ask. Okay. So I'm after mass of nitrogen. So mass of nitrogen equals N by V of HCl times molecular weight of nitrogen okay. I want percentage. Okay, so I want percent of nitrogen. So basically N by V of HCl times molecular weight of nitrogen over weight of the sample, however you weigh in your digestion tube, weight of the sample times 100. That gives me percentage. All right, so the volume here is the corrected acid volume. You're going to run a blank and you're going to run a sample. We always run a blank to correct for any nitrogen that can come from anything else you've used other than your sample. So the wax paper or weighing paper you use, the reagents you add. So the blank corrects for any nitrogen that is coming other than from your sample. So the corrected acid volume will be volume of sample minus volume of blank in milliliters. So we're going to titrate and it's going to be in milliliters. So usually normality and volume is in liters. So I want to convert milliliter to liter, so I multiply down here or divide. So this is divided by 1,000 to convert milliliter to liter, because your unit of the volume should be in liters. Because the units should be in liter, you divide by 1,000 to convert milliliter to liter. So that gives you an explanation for why there is 1,000 here. The 14 is actually your molecular weight of nitrogen, which is 14 grams per mole. Okay? And this here is going to be moles per liter, the unit of the normality. And usually we use a normality of 0.1 normal HCl that is standardized. The way you standardize the HCl in your previous lab, we did standardize or we haven't yet? I don't think we did standardization yet. That goes in pH and titratable acidity later. Anyway, we standardize to make sure we have the exact normality of the HCl so we can use in the equation with no error. So this is moles per liter and milliliter divided by 1,000 is liter. So the liters will cancel. Mole will cancel, and then the end is grams in 100 grams, which is percent. Any questions? I can repeat. Abby,
What's the extremely different?
the proton of HCl. And this is borate. So this should have -1, it's called thing. So this is borate ion. So this is an error. I should have -1 here. So this is ammonium +1, borate -1. So give you ammonium borate. So this should have -1. Okay, any other questions? Tyler?
What is the gram cancel out with the weight of the gram on top?
Well, that's grams per 100 grams of sample. So the percentage is actually gram per 100 grams of sample. So yes, it's a percentage, so it's canceled. Right? Yes, we have the grams, and then this is in 100 by 100 grams. Okay. Any other questions? I assume you know this. This is the time to ask. Okay, good. So bring your piece of paper that you're solving on, and I will collect it at the end of this lecture. So what you need to do is calculate rep 1 first, and then rep 2, and then get the average. I gave you sample 1 and sample 2. That means 1 replication of each. You're done? Yeah. Do you have a question? We'll have 17.5% nitrogen in the G protein. So you need to convert that conversion factor, which is 100 over 17.5 to get the conversion factor. Yeah. Yes, Tyler.
After you do that step, is the value we get in what instance?
No, it's actually a dehydrated sample, so it's on a dry basis, and then you want to convert to it. The other way around. Anybody with an answer? Not yet. Are we stuck? Is somebody stuck? Michael, are you stuck or? So you just need to apply this equation. Normality of HCl. What's the normality of HCl? 0.1142. We take the volume of the first sample, we subtract from it the volume of the next. So now you have the normality and you have the volume. You have the molecular weight of nitrogen is 14. You have the weight of the first sample, because we're doing one sample at a time, 1.015. And then you should get the percent nitrogen.
So that would be the 1.015 would be the weight of the sample on a dry basis.
On dehydrated, yes.
Okay.
The dehydrated sample. So your calculation is going to give you percent protein in the dehydrated sample, which you want to convert to wet basis. Anybody with a final answer? Tyler.
I don't know if this is right, but for sample one dry basis.
How much?
1.4%.
For percent nitrogen?
Crude.
There's something missing. Did you do normality 0.1142? Volume is 22 minus 0.2. Multiplication by 14. Divide by the weight of the sample by 1,000 multiplied by 100.
The
That is,
conversion factor is 100 over 17.5.
we do conversion last. You determine percent nitrogen. Yeah, and the conversion factor is 100 over 17.5. And you still got 1 point? Ah. All right, Madison. Nitrogen, and did you convert it?
Yes, I got 19.6%.
Okay. So what did you get? No? Okay, let's look at the math together. So you just apply the equation for first step. So your normality. This this is volume of sample. Listen to me. Michael, are you explaining it to Maya?
No, she's explaining.
Okay, so volume of the sample, the volume of HCl needed to titrate your sample minus volume of the HCl needed to titrate the blend. You multiply by the, sorry, there are no units here. You should use units. So this would be in milliliter. This is grams per mole. This would be mole per liter. This is in grams. And then you should get 3.43% for nitrogen. And then the 100 over 17.5 will give you the conversion factor. You multiply that by the percent nitrogen, you get protein. You do the same for sample 2. This should say sample 2, not sample 1. Oh, why am I seeing a lot of errors today? Okay. So you do the same thing for the second sample replication. And you get 2 reps, rep 1 and rep 2. All right, so you take the average and then you convert to wet basis. What's your wet basis situation? Anybody calculated wet basis? Yes, Abby? Yep. Okay. So you take the average and you have your average on dry basis because you said dehydrated sample. Then to convert to wet basis, you multiply by the weight of the dry. So that means 100 minus 8. 8 is the moisture content. Divided by 100, which is the weight of the total sample that is wet, on wet basis. So you have 8% moisture, then you have 92% dry solid. And you know that your 19.7 is in 92 grams of dry solid. How much you have in 100? So actually, no, that's not. Sorry, other way around. In 100 grams of dry solid, sorry. 19.7 is 100 grams of dry solid. How much you have in 92 grams of dry solid? So that's it. So you have 19.7 grams in 100 grams dry solid. How much you have in 92 grams solid? And that's how you get it. Okay. Any questions? Yes, please.
Yeah, when we driving, when you're just from up there, adding the 19.6, the 19.7.
These are rep 1 and rep 2 divided by 2. Correct. Ah. So you take the average from rep 1, rep 2 divided by 2 is your, yeah? Yeah, 100%. So 100 minus 8. So what is, this is telling you, let me erase this whole situation and explain that again. I'll have to redo this so that it has the unit, so it's not confusing. All right, so you have 8 grams moisture in 100 grams of sample, which is your pinto bean, okay? So you have 8 grams moisture in 100 grams pinto bean. The dehydrated sample is telling me now I have 19.7 grams. I have 19.7 grams protein in 100 grams dry matter, dehydrated pinto. Dry matter. This is the percent, 19.7% protein on dry basis. But if I want to calculate it on wet basis, originally I have 8 grams moisture. That means I have 92 grams of dry matter. So how much percentage protein I have in 92 grams of dry matter? So it's basically 92 grams, which is 100 minus 8, over 100 multiplied by 19.7%. So you get the 18.1%. Does it make sense? Now, we're converting dry to wet. In 100 grams of wet sample, you have 8 grams moisture and 92 grams dry. If you have 19.7 grams in 100 grams of dry matter, how much you have in 92 grams of dry matter? And that would give you that. Any confusion there? Okay. Dumas, which is another protein analysis method used to determine protein on nitrogen basis. So in this method, we combust the sample. So that's why it's called nitrogen combustion method, because here you're going to see that in lab, it's an automated automated method. You weigh your samples in little capsules, and there is a carousel here that you drop the capsules, and the carousel turns, and then the capsule drops into a furnace. So this oven here is heated about really high temperature, maybe 800 to 900 degrees C, and the sample combusts, meaning burns up. So there is, with the combustion, what happens is all of your sample burns, all of your matter turns into gases. So you have CO2, NO2, NO3, all oxides of nitrogen, carbon dioxide, CO2, and H2O. Everything combusts and everything turns into gas. So after combustion, all the gases goes to something we call a ballast. At the back of the unit, there is a compartment where all the gases are collected and flushed with oxygen. And from there, they go into scrubbers. So we have scrubbers that scrubs off carbon dioxide and scrubs off water. And from the scrubbers, they go to a place unit where they get reduced by copper. So cupric ions gets reduced to cuprous. And what happens is your NO2 and NO3 get reduced to nitrogen gas, to N2. So the nitrogen gas then, after being formed, it moves into the thermal conductivity detector. So thermal conductivity detector, same as the detector we use in GC, so it measures changes in conductivity. So as nitrogen reaches the thermal conductivity detector, the conductivity is converted based on the concentration of the nitrogen. Then you get percentage of nitrogen in the sample, and you get results in about 4 minutes. So this is another form where you have scrubber, but at the same time you have a column where the gases get separated, and you measure nitrogen content again using the thermal conductivity detector. So it's the same kind of compartment, but the one we have in the lab, it doesn't have a GC column. What it has is just a bunch of scrubbers to scrub off or remove carbon dioxide, remove water vapor, and just allow the nitrogen to reach the thermal conductivity detector. So basically, you use gases to combust at high temperature. The sample gets combusted, you have gases, and then the scrubber can be before or after the ballast. Here's where you get the mixing. And then there is a reduction tube to reduce oxides of nitrogen to nitrogen, and to the detector by thermal conductivity detector. So it's very simple, it's very fast. So if I want to compare Kjeldahl to Dumas, they're both based on calculating percent nitrogen and converting to protein content using the conversion factor. The difference, though, is that Kjeldahl is purely chemical reaction, Dumas is purely instrumentation. So in Dumas, you don't do much other than weigh the sample, put it in your little capsules, and then the instrument does everything for you. However, it's an instrument that requires a lot of maintenance. But actually running the sample, if the instrument is working fine and it's calibrated, it's really, really fast. It's very expensive unit. However, well, I want to say it continues to be expensive because maintenance is very expensive. So it's really high maintenance, costly equipment, but saves a lot on time. You have a lot of samples and the equipment is working fine. It's much, much faster than doing Kjeldahl. And it doesn't have any reagents, so there is no safety concerns there with concentrated H2SO4, concentrated sodium hydroxide. So you don't have issues here with using corrosive reagents. In terms of application, both are standard methods, official methods of analysis. They have similar accuracy and similar precision. You'll notice next week when you run a bunch of different samples, you're not going to see a lot of differences in terms of accuracy and precision. Well, if you know how to run Kjeldahl, you're going to get very precise data. And because Dumas is instrument-based, also it's very precise data. And in terms of accuracy, considering that we're looking at crude protein content, they both have similar accuracy. These are just to remember that these could be study questions. What's the principle of Dumas? How do we compare and contrast both methods? Another protein analysis method, which is secondary methods of analysis, it requires calibration. This is by NIR or MIR, mid-IR. So we're going to talk about IR spectroscopy on Monday, but basically in IR spectroscopy, we're measuring the absorption of energy by peptide bond, and specifically vibration of the energy, because it's this bond, it's the vibration of the bonds. IR is in the equivalent, IR energy is equivalent to vibrational energy. So it's very, very quick. However, it requires calibration. So it needs to be calibrated based on official methods of analysis. So it's a secondary method of analysis, and that needs to be calibrated using primary methods of analysis such as Dumas or Kjeldahl. But definitely it's fast, and if you're wanting to determine protein content during processing or on large set of samples, it's definitely a useful tool. For example, breeders, they have like meat breeders. They produce a lot of different breeding lines, and the need to screen protein content is very important for the price of wheat. So they have like hundreds of lines that they need to screen. So they calibrate an analytic instrument, and they can do screening very fast of the protein content of hundreds of lines. Okay, so there are other methods used for protein, but specifically research methods. And these, when we are isolating protein, or when we have pure solutions of an enzyme or pure solution of a protein that we want to test functionality on, we can do different types of methods. All are based on absorption detection. So light, basically spectroscopy. So a very common one we use in our lab is bisulfonic acid method, or BCA for short. In this method, it is based on reduction of cupric iron to cuprous iron by under alkaline conditions by the peptide bond acts as a reducing agent, and also the presence of cysteine, tryptophan, hydrogen. These amino acids can be reducing or can reduce cupric iron to cuprous iron. So the cuprous iron interacts with BCA agent to give you a color that absorbs light as 562 nanometers. In order to use this method, we have to always run a standard curve. And the assay actually comes, you purchase a PCA kit that comes with the reagent, with the cupric, with the BCA, with the cupric ion, and with a bovine serum albumin protein standard. So you prepare the standard at different dilutions. You react it with a different reagent, the cupric reagent, and then the BCA, and then you determine absorption, and you get the calibration curve with a prediction equation, and you use that to predict the protein content in your solution. Very simple and straightforward. Bradford assay, which you will be doing in the lab next week on a protein isolate or a concentrate that we purchase. So Bradford assay is based on the ability of the protein to bind a dye. So proteins will bind to chromatin blue based on their amphoteric nature, amphoteric meaning having the acid and base characteristic in their structure. They can bind chromatin blue, and the higher the concentration of the protein, the higher the concentration of the compound that will change from red to blue. The chromatin blue will change in color, and then you would measure absorption at 595. So again, it is sensitive, it's rapid, and it's mainly for proteins, not for peptides. And it has an acceptable outcome. You will be doing that in the lab and comparing the protein content you get to the protein label on that protein concentrate or isolate that we will give you. Last one here is measuring absorption at 280. We did say that the protein contain 3 amino acids, tyrosine, tryptophan, phenylalanine, that are phenolic amino acids. But mainly tyrosine and tryptophan have higher absorptivity than phenylalanine. So So it's basically the absorption of tyrosine and tryptophan at 280. And every protein has a certain amount of tyrosine and tryptophan, so all proteins absorb at 280. And also you can generate a calibration curve using a standard protein, such as bovine serum albumin is always used. It's non-destructive. You don't add anything. If you have the protein in solution, you can measure absorption and then reuse that solution. This is very, very helpful for us when we have limited amount of protein and we need to determine the amount of protein and then do another assay. So it's very helpful that it is a non-destructive method because you don't add any reagents. It's just the protein in solution as long as your solution is clear and it's not cloudy. Then you can measure absorption with minimal interference. So being pure and clear solution is very important. Pure, that means it doesn't have, the solution doesn't have other compounds that absorb light around the same wavelength. If you have other substances that interfere and absorb at 280, you'll get an overestimation of your protein content. So these methods, absorption at 280, the Bradford assay, the BCA, usually we don't use them for complex protein matrix samples. Because, for example, the BCA assay is based on reducing power. So if in your sample, anything in the sample has reducing power, it's going to reduce cupric to cuprous, and you get overestimation. So these assays are not really used to determine exact amount of protein in different sets of samples. They are very specific to research-based methods. So the other methods here, we have Duret and Lowry. They are very similar to BCA. They're just different use of the agents. Anyway, I think this is it. Happy Friday, everybody. I'll see you on Monday. Yes, now you can hand in your sheet for extra credit.
Quick question. So the lab report's due Monday now, with all the snafu data, correct? I just Caitlin asked, I just wanted to I was confused too, I thought it was today. Okay.
Yeah.
It hasn't been changed on Canvas. That was the big thing.
Send a note to Laura.
Okay.
I don't know if she's,
Okay, I get it.
yeah.